Good day, Mom. I am Archelan Delicano. So today, I will be discussing about plant pathogens as biocontrol of weeds and conventional approach. So first, what is plant pathogens as biocontrol of weeds and conventional approach? Plant pathogens offer distinct advantages over insects as biocontrol agents, primarily due to first, they are often more host specific, and second, they can be applied with conventional spray equipment at a time when the weed is at its most susceptible stage. A notable example of this approach is the introduction of Oxenia chondrilena in Australia in 1971, a host specific rust that effectively controlled the invasive skeleton weed and significantly reduced our reliance on herbicides. By leveraging the natural antagonistic relationships between plant pathogens, such as fungi and bacteria, and invasive weeds, biological control, it aims to reduce weed population through sustainable means rather than chemical interventions. Classical biological control involves the intentional introduction of these pathogens for long-term suppression, while inundative or augmentative biocontrol focuses on mass production and application of native pathogens for temporary control. Given the drawbacks of conventional weed management, such as herbicide resistance, biodiversity loss, and negative impacts on non-target organisms, integrating biological control methods with traditional practices that presents a promising strategy for enhancing weed management while minimizing environmental risk. Characteristics of plant pathogens as biocontrol agents of weeds. So, plant pathogens, particularly in fungi and bacteria, are utilized as biocontrol agents to manage the invasive weed species. Their effectiveness and characteristics can be understood through their mechanisms of action, interactions with their host plants, and the ecological context in which they can operate. So, here are the characteristics of plant pathogens in biocontrol of weeds. So first is host specificity. Many biocontrol many biocontrol pathogens are host specific, targeting only in a certain weed species while sparing native plants. This specificity is a crucial for minimizing unintended ecological consequences and ensuring that non-target species remain unharmed. So second is mechanism of action. And under sub mechanism of action, it has a pathogen induced manage. Pathogen can be significantly reduced with biomass, reproductive capacity, and overall population density through disease induction. For instance, fungal pathogens can cause tissues necrosis and disrupt nutrient uptake in host plants. Second is the mycoparasitism. Some fungi exhibit mycoparasitic behavior where they can attack other fungi that may be beneficial to the weed or that compete with biocontrol agent itself. This interaction can enhance the effectiveness of the biocontrol agent by reducing the competition or, or pathogenic threats. Third is the interaction with endophytes. Endophytes are microorganisms that live within plant tissues without causing disease. They can influence the effectiveness of biocontrol agents by either enhancing plant resistance to pathogens or combating with introduced pathogens. The presence of beneficial endophytes may sometimes hinder the establishment of biocontrol pathogens. Fourth is environmental adaptability. So, the success of plant pathogens as biocontrol agents often depends on environmental conditions such as climate, soil type, and the presence of other organisms. These factors can affect pathogen establishments and proliferation in the target environment. Fifth is the long-term efficacy. Once established biocontrol agents and can provide sustained control over invasive weeds, potential, potentially it leading to long-term reductions in weed populations without the need for repeated applications, which is often required in conventional herbicide use. And the characteristics of conventional approach, and it have three columns. So uh, first is aspects, second is plant pathogen biocontrol, and third is conventional methods. So in aspects, so under sa aspect, it have a methodology. In plant pathogenic biocontrol, it can utilizes in living organisms pathogens, and in conventional methods, it can release on synthetic herbicides. And second is the target specificity. So target specificity in plant pathogen biocontrol, it can highly specific to target weeds, and in conventional methods, it can often a non-specific affecting many plants, or it often non-specific affecting in many plants. And in uh, third is environmental impact. So environmental impact, it can generally uh, have a it can generally have an eco-friendly and sustainable. And in conventional methods, it can lead to environmental degradation. And fourth is long-term efficacy. It can have a potential for long-term control, and it often requires a repeated applications. And fifth is cost effectiveness. So under in plant pathogenic, ang cost effectiveness, it can it considered as cost effective over time. And in conventional method, it can initially or in it is an initial cost may be lower, but recurring cost can add up. So next is the parts and function of plant pathogen as biocontrol agents. So in plant pathogens, particularly in fungi, are increasingly recognized for their role in biological control or biocontrol of weeds. This approach leverages the natural interactions between pathogens and plants to manage invasive weed population. So, under parts and functions, they have a key components of plant pathogen in biocontrol of weeds. So, first is the fungi. The most commonly used plants pathogen for biocontrol are fungi. They can enter plants through wounds or natural openings, causing diseases by disrupting nutrient flow or producing toxins that impair plant growth. Second is bacteria. While less common than fungi, certain bacteria are also being explored for their potential in weed biocontrol. These microorganisms can reduce metabolites that inhibit the pathogenic fungi and bacteria, thus protecting plants from diseases. Third is virus and mycoplasmas. Though not as frequently utilized, some research indicates that viruses and mycoplasmas may also play a role in controlling specific weed populations. And under parts and functions, and in functions of plant pathogens in biocontrol, first is pathogen-induced manage. Once introduced, these pathogens can severely damage and target weeds by reducing their biomass, reproductive capacity, and overall population density. Second is host specificity. And in functions of plant pathogens in biocontrol of weeds, they have three types. First is the pathogen induced manage, second is host specificity, and third is ecosystem balance. Pathogen induced manage. These pathogens can severely damage and target weeds by reducing their biomass, reproduction capacity, and overall population density. And second is the host specificity. It can effectively biocontrol agents are typically host-specific, meaning they target only the invasive weed species without harming native flora. This specificity is crucial for minimizing ecological disruption. Third is ecosystem balance. By introducing the natural enemies of invasive weeds, biocontrol helps restore an ecological balance in the affected areas. This contrasts these conventional methods often rely on broad spectrum herbicides that can harm a non-target species and disrupt ecosystems. In parts and function of the conventional approach in plant pathogens as biocontrol of weeds, so the the conventional approach to using plant pathogens as biocontrol agents for weeds, it primarily involves the classical biological control. This method focuses on intentional introduction of host-specific pathogens that have co-evolved with target weed species. This method focuses on the intentional introduction of host-specific pathogens that have co-evolved with target weed species. They have a key components. First is host-specific pathogens. The use of pathogens that specifically target a certain weed species that ensures non-target plants are minimally affected. These pathogens include fungi, bacteria, and viruses that have evolved alongside the invasive weeds. They are meant to be controlled. Second is the establishment and purification. So, for classical biocontrol to be effective, the introduced pathogens must establish themselves in the new environment and proliferate over time. This leads to sustained pressure on the weed population, ultimately reducing its biomass and reproductive capacity. Third is the long-term control. Classical biological control, it aims for permanent establishment of the pathogens in the ecosystem, which allows for ongoing management of weed population without continuous human intervention. And in functions, first is the reduction of weed biomass. Second is decrease in reproductive capacity. Third is the ecosystem balance. And fourth is the integration with other management practices. So, in reduction of weed biomass, once established, plant pathogens can significantly reduce the biomass of target weeds, leading to lower competition for resources like water and nutrient among crops. Second is the decrease in reproductive capacity. Pathogens can impair the reproductive structures of weeds, leading to fewer seeds being produced. This is crucial. This is a crucial for long-term management as it helps to deplete the seed bank in the soil. Third is the ecosystem balance. By introducing a natural enemies of invasive weeds, classical biocontrol helps to restore ecological balance, and this method can reduce a reliance on chemical herbicides, promoting a more sustainable agricultural practices. And fourth is the integration with other management practices. Uh, the classical biocontrol is often part of the integrated weed management strategy that includes cultural practices, genetic technology, and other biological controls to enhance overall effectiveness against weeds. And also, they have a challenges. While classical control has proven effective in many cases, they have a challenges. It include first is agent establishment variability, and second is the potential non-target effects. So, in agent establishment variability, the success rate of introduced pathogens can vary based on environmental conditions and interactions with native species. In potential non-target effects, although it is designed to be host-specific, there is always a risk that introduced pathogens may affect a non-target species or disrupt an existing ecological relationships. Next is the classification. Domain in Eukarya, group group of fungi including the various pathogenic fungi and bacteria such as specific bacterial pathogens. and family. This varies widely depending on the specific pathogen being referenced. For instance, a common families include uh, Fusariaceae for Fusarium species and Sclerotiniaceae for Sclerotonia species and Xanthomonaceae for Xanthomonas species. And the genus, it is a specific genera of interest include Poxenia, Sclerotonia, and Xanthomonas. So next is the life stages or the life cycle of plant pathogens as biocontrol of weeds and a conventional approach. So first is the infection stage. So first is the infection stage. The life cycle begins when the pathogen such as fungus or bacterium comes into contact with the target weed. This can happen through spores or other reproductive structures that land on the weed's surface. And successful infection requires a sustainable environmental conditions such as moisture and temperature to facilitate the pathogen growth and penetration into the host plant. Second is the colonization stage. Once the pathogen infects the weed, it begins to colonize the plant tissue. This stage involves the pathogen growing and reproducing within the host, often leading to often often leading to symptoms like wilting, discoloration, or necrosis. The pathogen may produce enzymes and toxins that disrupt the plant's normal physiological processes, weakening it further. And fourth is the dispersal stage. The final stage involves and fourth is the dispersal stage. The final stage, it involves the dispersal of these reproductive structures to new hosts where they can initiate the cycle again. This stage is crucial for establishing the pathogen in new environments and ensuring its long-term effectiveness as a biocontrol agent. And fourth is the dispersal stage. The final stage, it involves and fourth is the dispersal stage. The final stage involves the dispersal of these reproductive structures to new hosts where they can initiate the cycle again. They can initiate the cycle again. This stage is crucial for establishing the pathogen in new environments and ensuring its long-term effectiveness as a biocontrol agent. In conventional approach to weed management using plant pathogens, these life stages are critical for understanding on how pathogens can effectively suppress weed populations over time. This establishment of biocontrol agent relies on its ability to complete its life cycle multiple times during a growing season, ultimately leading to the significant reduction in weed biomass and reproduction. In conclusion, using plant pathogens as biocontrol agents for invasive weeds, it presents a promising and sustainable solution for managing weeds in agriculture. This approach takes advantage of the natural relationships between these pathogens and their target weeds, effectively it controlling weed populations while minimizing the environmental risk that come with chemical herbicides. For instance, the success of Poxenia chondrelina in suppressing skeleton weed it demonstrates on how pathogens can help to restore balance to ecosystems and promote a biodiversity. As the farmers and agricultural practices increasingly focus on sustainable and combining biological control methods with traditional strategies will be crucial for building a resilient farming system and ultimately seeing the power of plant pathogen not only helps to tackle the challenges posed by invasive weeds but also encourages more environmentally friendly farming practices that benefit both the land and the future generations.